Hello again. Welcome back to Ms. Hook's Cardiovascular System lecture. We just finished this slide talking about the atrioventricular valves and how they're between the atria and ventricles on each side of the heart. The other set of valves you have are your semilunar valves, and these are between your ventricles and the artery they supply with blood. So they're these little like half moons, semilunar, these little cups, versus a cuspid here. There's a semilunar. And they're named after the artery that they're between. So the aortic semilunar valve is on your left side between the left ventricle and the aorta. So those prevent blood from going back into the ventricles after it's been pushed into the vessel. And the pulmonary semilunar valve is between the pulmonary trunk and the right ventricle, going to the lungs. Good. So this is from the view of the artery. So this is the aorta that we're in here. And underneath there is the ventricle. So when the ventricles contract, they push blood up through here, but when the ventricles relax, blood can go into these little cups, but it can't go back into the ventricle. So here's a view showing the cuspids. Remember, the right side's thinner. This must be my left ventricle. This is my tricuspid, bicuspid or mitral valve. And then here's a view in a ventricle. We've got a cuspid with the chordae tendineae, those cords connecting the papillary muscle to the cusp. Remember, tendons connect muscle to bone, so these connect muscle to cusps. If you have valve problems, it's called a heart murmur. You can hear the regurgitation of it going back, not closing completely. Okay, so to review, your heart has two sides. There are two pumps. The right pump goes to the pulmonary circuit to the lungs, where deoxygenated blood goes to the lungs. The lungs there, they drop off oxygen into the blood picks the oxygen off, and the blood drops off carbon dioxide and it goes into the air of the lungs, and we breathe it out. So we get oxygenated blood back to the left side, and then the left side pumps out to the body to the systemic circuit, where all of our tissues are getting the oxygen they need. So this is a really nice view. Deoxygenated blood is shown in blue. Remember, it's not actually blue, but it's a good representation. Deoxygenated blood goes from our body to the right side, out to the lungs, to the pulmonary circuit, where the blood picks up oxygen and becomes oxygenated, and then it goes back to the left side. The left side then pumps it out to the body, where the tissues, everything that needs oxygen, picks the oxygen up, and that blood is now deoxygenated and needs to go back to the right side. Okay, so what I would do now is pause the video, go back to this picture, and trace blood's flow. So starting out, let's say from the body going back to the heart, list all the structures, including valves, that the blood would go through. So it would be like the vena cavas, right atrium, through the tricuspid into the right ventricle, through the pulmonary semilunar valve into the pulmonary trunk and pulmonary arteries, into your lungs, through the pulmonary veins, back to the left side. Okay, so take a second to do that, and then check your work with these slides. Oh, and this is a really cool innovation that they found, where they have this what's called a continuous circuit of blood moving through. They don't have any sort of pumping, so it's all going through at once, and so you don't have any difference in pressure and you don't feel a pulse. Okay. Let's briefly discuss coronary circulation now. So your heart muscle needs oxygen, and the walls of the heart muscle are so thick that oxygen just can't diffuse and get to everything. We need a separate set of vessels. So coming off the aorta, you have the coronary vessels. So coronary arteries go around to the different portions of your cardiac muscle, get it all oxygenated, and then that blood that's deoxygenated returns to the coronary veins into a place called the coronary sinus, this big sac that empties into the right atrium. So you can see the coronary sinus on the back there, this thickened area, before it dumps back into the right atrium. So you have a lot of branching, these different alternate routes that blood can take. It's very important that cardiac muscle tissue not be deprived of oxygen. We need as much oxygen there as possible. If the cells don't get enough, they can die, and if they die, they become fibrous scar tissue. They don't regenerate. So we have all these different routes that blood can take. Those different routes are called anastomoses, these different branching points. If you don't have oxygen, if there's some sort of deficiency in your blood flow, but the cells don't die, it's called angina pectoris. If you have a heart attack, a myocardial infarction, there the coronary system has been blocked for too long, the cells from those vessels die. And then they're replaced with this non-contracting, this just scar tissue, fibrous, dense scar tissue. And kind of a funny one, the Grinch, his heart grew 3 sizes that day and he promptly died of cardiomegaly. That's an enlarged heart. What's really exciting is that scientists have been able to, in the lab, like in a petri dish, take cardiac muscle cells and give them epithelial growth factor. And those cells do divide and make new cardiac muscle cells. So that's an interesting area of research. Next, we're going to get into cardiac physiology. So we're going to look at how the heart sends electrical signals and contracts here. Okay, so electrical signals. We're going to start out with the pacemaker cells. The pacemaker cells are part of what's called an intrinsic cardiac conduction system. So intrinsic, it's inside the heart. It doesn't get outside influence too heavily. Cardiac heart, conduction, sending signal. So what happens, just like in nervous tissue, just like in skeletal muscle cells, is that our cells resting are at a negative charge. Our cardiac muscle cells are negative 60, whereas like our neurons were closer to negative 70. But each heartbeat, the pacemaker cells will have what's called a depolarization. They're going towards 0, and they get less negative. And once they reach negative 55 millivolts, that's the threshold, that opens the sodium-gated channels. Sodium rushes into the cell. Remember, the sodium-potassium pump makes this difference where there's lots of sodium outside the cell. So sodium diffuses in, and all this positive charge coming into our cell makes our cells more positively charged. Once you get to this point, that's an action potential. That electrical signal, that influx of positive ions, is going to spread to the next cells around it, to the next through those gap junctions, and spread. And each time the cell depolarizes, it contracts. That causes your sarcoplasmic reticulum, your endoplasmic reticulum, to release calcium. And then the actin combined to the myosin, your muscle cells contract. So each time the pacemaker cells send a signal, the heart muscle contracts. So each of these action potentials is one whole heartbeat. Starting here, going on. Second heartbeat, third heartbeat. After the action potential and they're done contracting, you'll see this repolarization where they go back negative again and get reset for the next heart rate. There's a great animation. Okay. So remember, these cells are self-excitable. They have this auto-rhythmicity. They're self-run. So those pacemaker cells are going to be repeatedly having these action potentials that go off, cause a heartbeat. Yes, they can be influenced by your nervous system, and they will be. The nervous system will make it easier or harder for them to depolarize, but they'll be continuously depolarizing on their own. So each time your pacemaker cells have an action potential, that propagates and spreads through the rest of your cardiac muscle for a heartbeat. How it spreads is a very specific route. So your pacemaker cells are in your sinoatrial node, your SA node. That's where the signal starts. And that signal spreads through the gap junctions into your right and left atrium. So the atria, the top chambers, get the signal first and they contract. blocking off the ventricles from the atria is that fibrous skeleton that we saw that holds up, anchors down the blood, our valves in our heart. So it's not going to spread down to the ventricles yet. What it does is it gets to the atrioventricular node and it pauses. Atria are contracting, the ventricles are filling. Then that signal is going to spread through the AV bundle to the bundle branches to the Purkinje fibers to the whole ventricle walls. And that signal happens really fast. And once those parts of your heart muscle get the signal, they contract. Okay. So let's go through each of these steps in more detail. So first you have your pacemaker cells in the SA node. Those are the ones that are going to be depolarizing and sending an action potential. They depolarize faster than any part of the cell, your myocardium, so they're the ones that determine your heart rate. If they're not working, other parts like your AV node do also depolarize, but they go slower. So if they get input from your SA node, they'll go with that. Okay, so your SA node has depolarized, sent an action potential. That spreads to the atrial walls. And then it moves from there to the AV node. And here, when we get to the AV node, you've got less gap junctions, smaller diameter fibers. So what we're saying there is we're basically going down from like 10 lanes of highway to 1. So we've got a lot of backup. We've got a lot of slowdown. Relatively large amounts of slowdown. We don't notice it, but our heart sure does. And so while that's paused, our atria are contracting. That gives the ventricles more time to fill. If there wasn't that pause, there wouldn't be enough blood in the ventricles, and it wouldn't be as efficient to pump our blood through. So that nice pause gives the ventricles time to fill. Okay. Then, going down into the ventricles, we go through the bundle of His, the AV bundle. That's the only way we have electrical signal going from the atria to the ventricles. Then that spreads to the right and left bundle branches, towards the apex, the point of the heart, and then the Purkinje fibers. And this is like going through the bundle of His. It's like going back to 10 lanes. So it goes slow, fast. We want all of our ventricle muscle cells to get the signal at once so that they all contract at once, so they have the strongest contraction. If half of the ventricle contracted and then the other half did, that wouldn't be nearly as effective as both parts contracting at once. So pause the video, go through those steps. Now we're going to look at the electrical signal that's read through this process. So we're able to monitor the electrical activity of the heart and see these different processes happening. See that signal spreading, those areas contracting and relaxing. Cool. So if your heart beats irregularly, that's an arrhythmia. And if it starts pumping really rapidly, these irregular contractions, the ventricles don't have time to fill, you're not going to be pumping blood properly. That's called a fibrillation. And in fact, the ventricles can start pumping so fast that they'll just stop. So when somebody on TV goes into VFib, that's ventricular fibrillation, and their ventricles have stopped. So what they do is they shock them so that hopefully they can reset the pacemaker cells, get the heart pumping properly again. So the heart activity is monitored with an electrocardiogram, ECG or EKG, they're the same thing. Cardio would be German with a K. And so this is going to monitor our whole heart at once. So you place these leads on the body so that they can take in the electrical information, and then you get this readout of the heart. And in the next video, we're going to go through this.